Good day, my name is Ekaterina Mironova. I am PhD, head of the Laboratory of Molecular Neuroimmunoendocrinology, St. Petersburg Research Institute of Pulmonology, and also senior researcher of Laboratory of Molecular Mechanism of Aging, St. Petersburg Institute of Bioregulation and Gerontology. Today we will discuss molecular mechanism of peptide regulation of the bronchopulmonary system functions. The World Health Organization classifies respiratory diseases as a priority, along with cardiovascular system diseases and oncological diseases. Respiratory diseases form very high levels of morbidity, disability, and mortality of the population, while remaining potentially preventable. However, drug treatment in many cases doesn't lead to positive results since it is carried out with the help of drugs that are aimed at eliminating the cause of the disease at best and at worst act only symptomatically. Thus, many of the drugs have side effects, negatively affect the gastrointestinal tract, are contraindicated in liver failure, myocardial infarction, arrhythmia, and epilepsy. In a significant number of cases, their application leads to the development of allergic reactions as well as nausea and vomiting pain in the epigastric region. It should be noted that at present there are particularly not medicines aimed at restoring the function of the lung tissue and bronchial mucosa after the development of the pathological process, regardless of the reasons that caused it. In this regard, the development of new medicines used to treat respiratory diseases is becoming increasingly socially important. Naturally occurring substances are of greatest interest, taking into account their safety and lack of toxic effect. Such medicines include peptides, isolated primarily from the bronchial mucosa. The tripeptide congluten was synthesized from the polypeptide complex taxarest, which was isolated from the bronchial mucosa of young animals. In a study of dissociated cultures of red bronchial epithelial cells, the peptides congluten reduced the expression of cellular aging markers P21 and P53, thus normalizing the functional activity of bronchial tissue. Effect of congluten on P53 expression in bronchial cell culture of passages 3 and 14, where A is control passage 3, B is congluten passage 3, C control passage 14, and D congluten passage 14. The tetrapeptide AEDL was also synthesized from the polypeptide complex taxarest, which was isolated from the bronchial mucosa of young animals. The effect of IEDL peptide on differentiation of human embryonic lung cells was studied using quantitative polymerase chain reaction. On the left, you see the control group. On the right, you see where is IEDL peptide was added. It was found that IEDL peptide promotes increased expression of the genes which involved in the differentiation of bronchial epithelium. You can see our results on the graph. The tetrapeptide IDL specifically regulated the processes of cell renewal in the bronchial epithelium through the effect on the proteins expression and the functional state of the cell through the effect on the protein's expression also. All results you can investigate on our graph. On this picture on the left you can see the control group, and on the right you can see the cells with ideal peptide addition. It was established that the use of tax arrest in addition to standard therapy in patients with chronic bronchitis in 82% of cases contributed to an improvement in well-being, a decrease in the frequency of coughing attack, asthma attacks, and a decrease in amount of sputum. Positive dynamics of indicator in the control group was noted and in 57% of patients. Auscultation of the lungs in dynamics indicated a decrease in severity of dry wheezing. Tax arrest contributed to the improvement of external respiration indicators. After standard therapy, in combination with tax arrest, the vital capacity, total capacity, and forced expiratory capacity of the lung increased compared to the indicator before treatment. All our results you can see on the table. The molecular mechanism of biological activity of the polypeptide complex tax arrest is associated with the ability of short peptides EDG, which is congluten, and also peptide IDL, which are part of tax arrest. This regulates the synthesis of a wide range of proteins in the human bronchial epithelium. The tripeptide congluten and tetrapeptide IDL specifically regulate the expression of genes involved in the differentiation of bronchial epithelium and also normalize the processes of cellular renewal and the functional state of bronchial cells. In addition, the polypeptide complex tax arrest is an effective and safe medicine in the complex treatment of patients with chronic bronchitis. The study of the molecular mechanism of aging of peptides of the bronchopulmonary system allows us to recommend their use in case of dysfunction of the lung and bronchi in diseases of the respiratory system of various origins, as well as to maintain the function of the respiratory system. Thank you for your attention.